It's great to see all of you here. Today we're going to be discussing how errors propagate in spatial biology, specifically in digital pathology. But before we get started, a bit of an introduction. Like Noach said, my name is Becky Arviv. I am originally from Atlanta, Georgia. I studied biomedical engineering at Duke University, where I did research in high-frequency ultrasound. And I moved to Israel about 2 years ago and have been working as a machine learning engineer at Nucleai ever since. So before we get started, let's outline what we are going to discuss today. So first, I'll introduce digital pathology. For those of you who are not familiar with our domain, I'll give you a brief introduction so we're all on the same page. We'll then discuss some of the challenges and limitations that we face as part of our workflow in digital pathology and in medical imaging in general. We'll then discuss how exactly errors propagate through this workflow, and then discuss some strategies and suggestions that we can use to manage the errors that we see that exist and propagate throughout our data flow. Okay, so first of all, the basics. What is pathology? So first of all, a biopsy is taken, and pathology is a study of distinct cells and tissue organizations within our biopsies, allowing us to diagnose diseases and evaluate prognosis for patients and for treatment selection. Pathological evaluation is the standard of care. It's the gold standard for diagnosis in many diseases such as cancer. And so let's discuss what the typical workflow is in a conventional pathology. A patient will arrive at a hospital. They'll have their biopsy taken by a physician. And this is a piece of tissue that is then transported to a laboratory, at which point it is processed, which includes several steps that include slicing the tissue very, very thin, staining it with any number of stains so we can see different aspects of the cellular environment, transporting it back to wherever the pathologist is physically located, at which point they'll analyze and view the slide under a microscope. When they're finished, they'll physically store the slide for later use. So how does digital pathology come to disrupt this workflow? Once the tissue is cut and placed on our glass slide, rather than physically transporting it back and forth to the pathologist and analyzing it in an analog way, we can digitize the slide and we can upload it to a database or a file system, which allows us to not only have the slide analyzed by pathologists anywhere in the world at any time, but it also allows us to use tools like artificial intelligence to improve the accuracy of our analysis and extract more quantitative insights from our slide. So let me explain what I mean. On the left here, you can see what to the untrained eye might look like a nice marble image. But to a pathologist, you might be able to look at this and give a certain diagnosis. They might be able to tell if our patient has cancer or does not. With some more analysis, they might be able to tell you if it's grade 1 cancer or grade 4 cancer. However, what they're not going to be able to tell you is exactly how many cells we have here. They're not going to be able to tell you exactly what the density of our cells here or what the spatial relationship between the cells are to each other. And the great part is that computers actually are really good at doing this. On the right, you can see what our goal is, our objective in what we achieve when we use artificial intelligence to create higher level structure to our slide. Not only is it interpretable and understandable to a lay audience, right? All of us may not be able to understand what is happening in the image on the left, but we can kind of see that there are certain cells that are identified here in certain areas, etc. But why bother? What do we gain from going digital? Pathologists could look at a biopsy and give you some sort of, again, a qualitative analysis, perhaps saying that we see an immune response or that this patient has cancer. But they're not going to be able to tell you exactly how many immune cells we see or some more quantitative predictive factor. This is going to allow us to improve clinical decision making, and which leads to better patient outcomes. Furthermore, as I mentioned before, we are now able to have our slide analyzed by pathologists anywhere in the world. This really opens up the possibility for remote medicine and lots of other opportunities in that area. Now, this is on the clinical side. On the research side, we also gain a lot of valuable insights from going digital. A pathologist might, for example, struggle to look at a slide and say which drug, for example, might be better for a patient with cancer. Let's say our drug works well for patients that have a certain cell type next to their tumor cells. a pathologist will have to go manually and select patients one by one for this trial. This includes a lot of manual work. However, once we introduce digital pathology, we can have this selection done in a very streamlined manner, accelerating drug development, clinical trials, and again, thus improving patient outcomes. Now, let's go into a bit more detail of what this workflow looks like. So, for example, here at Nucleai, once we acquire our data, we have files that display images of our tissue. Okay? Once we acquire our data, the next step is to annotate it. This includes identifying certain cells or certain areas and having expert pathologists identify these key features. The next step is to input these annotations into a deep learning model, at which point we'll output the result you see on the right. We can then input these structured results into a feature calculator. We can then calculate high-level features that are going to give us more insights into what we see in our slide. And then we can output those into a statistical model to make any sort of prediction. Perhaps we want to look at how responders to a certain drug look versus non-responders. Or perhaps we want to give a diagnosis. We want to classify tissue as cancerous or non-cancerous. So let's go step by step into this process. So now you have the POV of an annotator here at Nucleai. And you can see that this is a very time-consuming and laborious process. It requires expert knowledge, and annotators are required to go one by one to identify certain cell types based on their morphologies, based on colors. And we'll discuss a lot more about annotations throughout the lecture, but it's important that we understand the difficulty and the pain points that arise with this step. The next step is to input these annotations into a deep learning model. This step takes our annotations, we split them into a train and a test group. We can then train our model, depending on our annotations, to detect certain areas or detect certain cell types, which then brings us to our next step, a feature calculation. So here we can take our outputs from our cell model and better understand the complex relationships that we see between different cell types in the tumor microenvironment. We can perhaps look at different densities, different certain subregions within our tumor microenvironment, or even higher level graph-based features that really explore the complexity between these spatial interactions. The next step in our workflow is to take these calculated features and input them into some sort of statistical analysis. This can be anything from doing a simple EDA to see if we have different groups based on our feature values, or even some sort of predictive modeling to see if we can find a feature that separates in our data set between patients who respond to immunotherapy in our case or don't. And that's it. Lecture's over. No. If everything went as smoothly as I described there, we would be done and our workflow would be quite simple. However, in practice, the process I just described contains several points where errors can be introduced into our pipeline, and which adds lots of challenges to our analysis, and also makes our job a lot more interesting. So starting from the data acquisition, we have different errors that can arise, such as artifacts from our images, lack of standardization, and heterogeneity in our data acquisition. And this can also lead to the next input into our pipeline, which is our annotations. This can lead to annotator biases or ambiguities in our data points. Now I'm going to discuss a bit more in detail a few of these errors. So the first thing I'm going to mention is heterogeneity and lack of standardization. I want to remind you all, if we went back to our initial workflow, that the way our data is acquired is quite a manual and physical process, right? We're taking tissue from a patient, we're transporting it to a laboratory, we're slicing it, we're fixating it, we're staining it with different colors, and depending on a lot of factors along this pipeline, it can result in data that is quite heterogeneous. For example, depending on the scanner, the manufacturer of the scanner, we can have data that simply looks different than another manufacturer, or perhaps the company that makes the stain. That can also cause heterogeneities. So here on the, what side is that for you guys? That's the left. On the left, you see that we have several different images from H&E stains. H&E is a very common stain for us in pathology. And theoretically, we should be looking at the same images here. But due to the lack of standardization in different steps of this process, we actually see very different images. And the problem here is that the differences in staining and the differences in our image can cause different classes to be confused. For example, a dark stain might make all stromal cells look like lymphocytes, when in reality there are several different subgroups. There's lymphocytes, fibroblasts, etc. Next, we'll discuss artifacts. This is constant in any sort of imaging, specifically, especially in medical imaging. And this can be anything, in our case, from issues in our microscopy for tissue that is out of focus, or perhaps our tissue has folds. And again, these are all due to the fact that we must understand, and we must, as data scientists, take into account that our data doesn't just appear on our screen, right? It is acquired through a process, and depending on what our use case is, the process might have more or less errors along the way. All of these things build up to the fact that pathologists don't agree, right? We can look at lots of different high-impact papers that really display this in a more empirical manner, but depending on the task, we can have anywhere from 54 to 80% annotation agreement rate. So this is even looking at experts, looking at high-quality data, and even then, they still don't agree on the final diagnosis. And let's keep this in mind as we start discussing annotations down the pipe. So now I'm going to, now that we kind of understand a bit more about what our domain is and some of the problems that are going to arise in our data acquisition, let's see how these errors propagate through our workflow and through our pipeline. So we'll start by answering, we'll discuss this by exploring a few different questions. First of all, how can we model our errors in our annotations? Okay. Next, we'll see how the errors that we have that exist in our annotations are then propagated into the model training, how they affect the model's performance. Once we understand that, we'll see how the output of our structuring model, our model that gives us different cells and their locations, how does this affect, how do the errors here affect our feature calculation, our feature accuracy? So let's start with the first question. In an ideal world, we would use a different modality with a higher accuracy to create our annotations. But this is a kind of a cop-out, right? If we were able to use a higher quality method to create annotations, we wouldn't have manual annotators. However, many fields in medical imaging actually have other corollaries, other modalities that are higher quality. And the reason that we don't use these for our annotations in our model is because it's not always available, and B, it's quite expensive and not necessarily practical in a clinical setting. So what I suggest here is rather than using ground truth annotations, high quality ground truth annotations to train our models, which is not necessarily realistic, we'll use them to evaluate our annotators. So in our workflow in digital pathology, we actually have a way to do this. On the left, you can see an H&E slide, which like I mentioned, annotators will distinguish between different cell classes based on morphology or based on colors. However, we have a technology called multiplex imaging, which essentially uses different protein-specific antibodies that bind to specific cell types, so which have a much higher precision and recall than our manual annotations on our H&E slides. And so we can use this relationship between some higher accuracy, but let's call this ground truth, and our manual annotations that are created by our annotators. So to do this, let's go through a toy example. Imagine that we have an annotator with a given accuracy. So this annotator, he or she performs at about 80% accurate. 80% of the time, they give the correct annotation. And so if we looked at how this annotator's annotations are input into our model, you can see that if our annotator performs at 80% accuracy, the reliability of our annotations that go into our model are also at 80%. Pretty simple. But what we can do is we can model how, as we increase the number of our annotators, we can see how our majority agreement or our model accuracy of our annotations increases. So let me elaborate. Let's assume, and again, a lot of these assumptions are not very practical, but help us to kind of wrap our head around this type of problem and how we're going to explore it further. Let's assume that we have several annotators who all annotate with the same accuracy. So if I had 3 annotators, they all annotated, let's say, at 80% accuracy. And let's also assume that their mistakes that are completely independent of each other. They each tag at 80% accuracy, completely independent of the other annotators. Now, we see that when we only have one annotator who annotates at 80% accuracy, our model receives annotations that are 80%. However, let's say we increase our number of annotators to 3 and take the majority vote between 3 of them annotating at 80%. We now can derive or simply look at the graph to see what our majority vote accuracy is. We can essentially just take 1 minus our our error and then raise it to the power of the number of annotators. So if we had 3 annotators, our majority accuracy would be 92%. And now we see that we have this range that develops between our 1 annotator and our multiple annotators. But like I mentioned previously at the beginning, it's very clear in reality that our annotators are not completely independent of each other, right? A data sample that is hard for one annotator to annotate, if it's a difficult example, is very likely to be hard for the annotator next to them to annotate as well. So we can't assume complete independence, but let's also assume that they're not completely dependent on each other. If they were completely dependent, meaning that they made the same mistakes, everyone made the same mistakes, we would be back to the situation where we have 1 annotator, right? We would get back to the situation where we have 80% accuracy going into our model. However, because we know we're somewhere in between complete dependence and independence within our annotators, we're in this range between 92 and 80%. So this gives us a better idea, if we have some sort of ground truth, to understand where we fall on the spectrum. Now, because not all use cases and fields have some sort of higher quality ground truth to utilize, we can use annotator agreement as a proxy here. So let's assume we have some sort of relationship, some underlying relationship between the annotator agreement and our annotation accuracy. To clarify, I mean that as more annotators agree, you would expect that to mean something in terms of the ground truth reliability of our annotation. And this is more just to kind of set up this like thought experiment in your mind to think, how can we in our fields model our relationship between annotator agreement or any other proxy for our relationship between our annotations that we have in the real world and the ground truth that is not always achievable. So one of the methods we can use to measure our annotate agreement is a statistic called Cohen's kappa. This is one of many. There are many different statistics out there that essentially do the same thing. They look at how we can measure our inter-rater agreement. And what's nice about kappa is that it takes into account the observed agreement that we would expect by chance, right? So let's derive this equation. First, let's start by looking at P naught, which is our observed agreement, right? It's where both annotators annotated yes, and they both annotated no. We can then look at PS, which is the probability that both annotators will answer yes by chance, and likewise for P no, the probability that they'll each answer no by chance. PE is then the sum of P yes and P no, and then we can use both of these equations that we've derived and come to our Cohen's kappa, which essentially, as we see, is the observed agreement minus the chance agreement. And again, this is just one of many different metrics that can be used to better understand what the relationship between our annotators' agreement are. So now that we have a better understanding that our data that goes as input into our model is not 100% accurate, we can see how this propagates into our model training. Now, let's look at this toy data set here where we can see let's assume we have 2 classes, the yellow class and the green class. The red squares here represent areas where our annotators annotated incorrectly, which we saw previously is not only possible but very likely. We can then see on the right the output of our model, where check being a correct inference result and X being an incorrect inference result. And we can see that our model was incorrectly penalized for cases where it was actually correct, and the opposite, it was incorrectly rewarded for cases in which it was incorrect. So with this in mind, let's make a little simulation. So again, we're going to use a toy data set here that essentially has a lot of assumptions and is more so to get ourselves in the mindset of thinking of how we can apply this to our real world problems. So using a toy data set, we trained a simple classifier. The red line represents the accuracy that we achieved when we had our perfectly clean data set. We used ground truth for our training data set and ground truth for our testing data set, and we achieved an accuracy of approximately 77%. Now the experiment that we do here is we say, let's simulate our noisy labels that we have in the real world. So let's train our data set on noisy labels and evaluate it on a test set that's using the ground truth. And as you would expect, we see a decrease in model accuracy. Now, another interesting twist on this that we could do is we could do the opposite. We could train our data on clean ground truth data, and we could evaluate it on noisy data. Okay? So I want to point out here to maybe clarify that the red line, essentially modeling our perfect model, And our orange line, which is representing our model that was trained on clean data but evaluated on noisy data, are essentially the same model. The only difference in them is they're evaluated on different data. So we can see that our observed model accuracy is dependent, very dependent on the accuracy of our annotations, which again is a logical point to come to, but again takes some thought to get to. So in green we see what happens in the real world. We have noisy data in our training set and in our test set, and we can see we suffer both from the noise that's in our test set and in our training set. So again, there's lots of different tweaks that we could do on this experiment to make it more realistic. Another interesting experiment that we could do is add a validation set to this experiment and see how hyperparameter optimization with noisy labels affects our model accuracy on a ground truth test set. Again, lots of different ways we could play around with this, but just to get ourselves in the right mindset here. So lastly, what we can do is take the insights we learned from our model training and see how our errors from our model propagate into our feature calculations. So in order to do this, I'm going to present a sample problem from our domain. We're going to look at tumor-infiltrating lymphocytes. So for those who aren't familiar, lymphocytes are a subclass of immune cells that specialize in the identification and removal of pathogens in our tissue. They're able to recognize and kill tumor cells by identifying them as foreign cells, right? Essentially what they're doing is they identify cells that are not not part of the host tissue, and they're able to then recognize and kill them. However, our tumor cells are a bit smarter than that, and they have sort of several mechanisms that they use to evade detection. And in the development of immunotherapy, we can see how immunotherapy works by alleviating these mechanisms that allow for the reactivation of lymphocytes against tumor. It essentially helps our lymphocytes better detect our tumor cells. So again, let's refocus onto what we see here, tumor-infiltrating lymphocytes, so lymphocytes that are specifically in our tumor area. Why do we care? This is one of the major biomarkers for immunotherapy response, and it has been proven to be a positive prognostic biomarker in multiple indications, ranging from different types of breast cancer to lung cancer, ovarian cancer as well. And you can see here that from this graphic, as we have more infiltration of immune cells into our tumor, our outcome increases. So with that in mind, that background in mind, let's create another toy data set where we have 100 patients, and we divide them into two groups. We have group 1, which has a high survival rate. Their survival is over 4 months. And we have group 2, which has a low survival rate. Their survival as a group is less than 4 months. And so each group has 50 different patients, and each patient has 5,000 cells that we can run inference, and we can classify as either tumor cells or lymphocytes. Now, once we run inference, once we look at our ground truth from our data, we can see that we have some distribution of lymphocyte cells, immune cells, and tumor cells in our tissue. And using this, we can create a feature. We can look at a feature that is used in pathology today, the TIL ratio, the ratio between the number of lymphocytes in your tumor area and the number of tumor cells. And we can use this as our feature to model this problem. So if we calculate our feature, we see that this is a great feature to separate between our groups. We see group 1 has a high ratio of immune infiltration, and group 2 has a lower ratio, meaning group 1 has far more immune cells, almost double the amount of immune cells as group 2 does in the tumor area. Now, this is on ground truth data. This is assuming that our model worked perfectly, that we had no errors in our annotations, and we received 100% accuracy. But in reality, our model gives results that sometimes confuse tumor cells for lymphocytes and lymphocytes for tumor cells. So our inference when we run on our data is not going to be 100% equal to our ground truth. So let's look how our model accuracy affects our feature distribution. So on the top you see what we started with. This is our feature calculated with our ground truth. And on the bottom you can see after we've applied this non-perfect accuracy, we can see some changes in our feature. First of all, let's note pretty obviously that our feature has shifted along the x-axis. And another note is we've gone a bit closer together. We still have two very distinct populations. However, our feature has shifted a bit, and they're a bit closer together. So let's maintain the same error rate, but let's imagine that our model performance was actually dependent on our group. let's imagine that group 1 has a model accuracy that's slightly different than group 2. And again, in our use case, this can occur for a variety of reasons. For example, group 1 came from one lab and group 2 from another lab, or group 1 was annotated by one annotator and group 2 by another. And we can see how this might affect our feature. So you might see a very drastic change from the slide we saw before. You can see that our populations have moved closer together, which is starting to not differentiate our groups quite as much. To really drive this point home, let's do a bit of an extreme example here, where we can see where our model is confused basically in the inverse way. So group 1 detects 6% of its tumor cells as lymphocytes and 10% of its lymphocytes as tumor cells. And you can understand group 2 from the confusion matrix below. But now we have a model accuracy that's quite high, right? These two confusion matrices average to the same matrix we saw here before. These two differences could be hidden if we evaluate only on our data set as a whole and not between these two subgroups. Now, I'll make a few points here. One, this feature is quite ripe for this type of confusion. One, because if you look at the actual ratio, you probably can't see, but it's quite low. We have far more tumor cells than we do lymphocyte cells. So the errors that we get when we convert tumor cells to lymphocytes affects our model much more than the mistakes in the other way around. So that's one factor to be aware of. And then another is that we see that our feature that once was a clear separator between our two groups has now disappeared. So now we've seen a few demonstrations or a few thought experiments to explain how our error propagates down the stream. So now I'm going to suggest a few different solutions, a few different ways that we can help handle these errors. So first, if we remember our example with data annotation, we can use techniques of annotator weighting. Now, I use this example because now that it's easier now to train deep learning models. There's lots of crowdsourcing on the internet. AWS actually, for their mechanical Turk service, allows crowdsourcing for annotations. And in order to understand that and validate that our annotators are performing at a high level, we actually can evaluate our annotators against a ground truth, like we described, and then weight our annotators based on how well they perform. This allows us to weight annotations of higher quality, and then have expert annotators influence our training further. Next, we'll see how the errors in our annotations, how we can provide some solutions to the problems of annotations affecting our model. So first of all, in our model training, there's lots of different techniques here, but I'll speak quite generally. I'm not going to go into too much detail. We can use loss functions that are robust to noise, loss functions that expect some sort of weighting or know how to deal with noisy labels. There's a lot of work in the field specifically to describe these different loss functions, and like I said, I'm not going to go into too much detail here. Another method we can use is label correction methods. So this includes any sort of unsupervised or semi-supervised technique where we can cluster our labels together and flag, for example, labels that are data points that cluster together but have different labels. Now, looking at our model evaluation, we saw in a few examples how just in the way we evaluate our model can change the way we understand its performance, right? So one suggestion I have here is to try and evaluate our model not as an average across our whole data set, but try to evaluate between different possible confounding variables to understand if there's any underlying feature in our data between the confounding variables that will result in a decreased or a different model performance between our two groups. And again, this is difference in model performance in our structuring section. This is just identifying where our cells or areas are. This is even before we calculate our features. The next suggestion we have here is to invest in high quality annotations in our test set. Now, whether or not we have some sort of ground truth to base this off of, we saw before how our test set can make our accuracy look significantly lower or perhaps even higher, depending on irregardless of the model that we trained on, right? We saw before that our red dashed line that has our perfect model gave us a different model accuracy than our model that was the same, however, with different test data. And one way to do this is to invest and have multiple annotators label our data and use a majority voting method to ensure that these data points are of higher quality. Now, lastly, let's discuss how we deal with the errors in our feature calculation. So here we can try and design features that are robust to the noise and imperfect accuracy that we expect in our model. One way to do this is try to represent the same biological information in different ways to ensure that our model is not overly dependent on a single feature representation. In our example, we looked at the TIL ratio. Essentially, what we were trying to understand was not the number of lymphocytes divided by the number of tumor cells, but rather how much infiltration of immune cells do we have into our tumor. Another way to represent that biological characteristic is to look at each tumor cell and see what's the probability that it's near an immune cell. That's another metric, another feature that we can use to represent the same biological truth, but using slightly different representations of our data. Another suggestion is to really utilize our domain knowledge. This is kind of an obvious thing that data scientists hear all the time, but really it's worth drilling in another point to understand which features we want to calculate and which ones make sense in our given domain. So let's summarize what we discussed here. First of all, high quality annotations are critical. Again, this is kind of an obvious point, but really important that we drill through that annotations sometimes are seen by data scientists as not part of our model. But what we learn here is that it's really essential to understand where our annotations come from and to handle them properly because they will propagate downstream through our analysis. Next, I want to point out that another quite obvious fact that annotations in medical imaging are not perfect. And this is a fact that's shown in various forms of medical imaging between radiology, pathology, etc. And we should take this into account as well. Next, we really should try and utilize the tools that we have to better understand our annotation quality. Right? A lot of times, again, as data scientists, we assume our annotations are ground truth. But we know because given the first 2 points here that they're really not. And if we take into account this into our workflow, we can really improve our model training and downstream feature calculation. Next, there are methods that we can use in the training of our model to better handle noisy labels. Again, I didn't go into too much detail, but there exist lots of different methods in different loss functions and different data weighting parameters that we can use to better train our models. And lastly, we can work hard to engineer our features to be robust to noise and the imperfections that we have in our data. So thank you all for listening.